Hello friends, welcome to Binary Tips. Today we are going to see NCERT Class 10 Chapter 8, How Do Organisms Reproduce? So in this chapter I'll be dealing with the concepts of asexual reproduction and previous year's questions, and I'll also tell you how many marks does the question carries and how the question can be given in the question paper. So let's get started, alright? Now, the first question that arises is, what is reproduction? So what do I mean by reproduction in general? So reproduction means the production of new organisms from the existing organisms of the same species, okay, is known as reproduction. So every organism on this earth has the capacity to reproduce, okay, in order to continue its population or in order to continue the species number. Now, how is this process of reproduction helpful? So there is one more question that arises. How is the process of reproduction helpful to the organism, or significance or importance of reproduction? What is the importance of reproduction? So the first point is it ensures the continuity of life on Earth, alright? So only with the help of reproduction it is possible for an organism to continue its life on Earth, which means that all organisms need to reproduce to maintain their number, to maintain the population. Next, it is essential for the survival of species on Earth. So without reproduction, the survival of species on Earth is impossible. Okay. Do organisms create exact copies of themselves, or what is the importance of variation? So these are the two questions that is often asked for 1 mark or 2 marks. Do organisms create exact copies of themselves, or what is the importance of this variation? Okay. So to understand this, we need to understand that in our body, every character, for example, hair color, eye color, is stored in the form of information in the DNA. Okay. Now when this DNA, during the time of fertilization or during the time when reproduction happens, a copy of DNA is sent to the offspring, right? So every offspring carries one DNA, that is one set of characters from the mother and one set of characters from the father. So this DNA is passed on to the next generation when a copy is produced. I mean when the copying is taking place or reproduction is taking place. Now, is it exactly very similar to what the parents have? No. There are certain variations that take place during this process of DNA copying. So you can see the two strands of DNA are very different from each other, right? They are similar to the parent strand, right? The structure is similar. The basic design of this DNA is very similar to that of the parent, but it is not identical. Okay? The strands which are formed are not identical, which means there are slight variations in the DNA. We all don't look exactly like our parents, right? So there are certain variations. So these variations happen during the reproduction process. Okay, that is during the gametes when the fertilize, during that time this take place. Okay. Now, how is this variation helpful to an organism? How this variation serves as an importance to an organism? Now, let us consider with the examples of certain bacteria. Now, this bacteria starts reproducing and produces large amount of bacteria. Now, you can see that there are some bacteria produced with some sort of variation. Okay. Now, let us say that these bacteria that are formed are heat-resistant bacteria. Okay, the bacteria that are formed are heat-resistant. Now, if a situation occurs that there is global warming, there is excess of global warming on this earth, which means that the temperature of the earth rises, right? So at this condition, these bacteria which are heat resistant will survive, okay? Because they have the capacity to sustain so much of heat, whereas the other bacteria which were not heat resistant will die. Okay. Now, similar example is given in page number 128 of your textbook. So thus, if there were a population of bacteria living in temperate waters, and if the water temperature were to be increased by global warming, most of these bacteria would die. But the few variants resistant to heat would survive and grow further. So variation is helpful for the survival of species over time. Okay. Moving forward. So, types of reproduction. So, when I say types of reproduction, we have two types of reproduction. One is asexual reproduction and we have sexual reproduction. Asexual reproduction means the production of new organisms from a single parent without the involvement of sex cells or the gametes. And sexual reproduction simply means the production of a new organism from two parents by making use of sex cells or the gametes. Gametes means the sperms and the ova. Okay. Now let us understand the differences between asexual reproduction and sexual reproduction. Asexual reproduction, as we all know, does not involve the fusion of male and the female cells. There is no involvement of gametes, whereas sexual reproduction is involved in the fusion of male and female gametes. It includes the fusion of male and the female cells. Asexual reproduction, the offsprings produced are clones of the parents. They look exactly like the parents, but slight variations will be there which are negligible. Whereas in sexual reproduction, offsprings produced vary from the parents. Why? Because two parents are involved. Okay, and we are getting characters from two parents. So that is the reason that in sexual reproduction, the offsprings produced are very much different from their parent. They have the similar body design, but there are certain variations. They are not identical. They are not clones of the parent. Okay. Under asexual reproduction, we know that only one parent is involved. Okay. Sexual reproduction, two parents are involved. These 3 points you can write along with examples. Okay. So we'll see the examples in the following slides. This question can be asked for 2 marks. Okay, differences between asexual and sexual reproduction along with examples can be asked for 2 marks. So make sure that you write at least 4 points each. 3 points plus 1 example will do. Okay. Moving forward. Let us now discuss about asexual reproduction and its types. Now, what is asexual reproduction? It means that only one parent is involved and there are no gametes involved in this process. So, the first type of asexual reproduction is fission. Now, the word fission simply means to break. Okay? It simply means to break into 2 pieces. In the process of fission, an unicellular organism splits or divides to form 2 or more organisms. Now, fission is of 2 types. One is binary fission, where you can see the example of amoeba. So, amoeba, as you can see, first you can see that the nucleus goes on dividing and then the cytoplasm divides, forming 2 daughter cells. So these cells that are formed at the end are called as the daughter cells. So this is in case of amoeba and leishmania. Okay. So in leishmania, the process is a longitudinal one. Here it is horizontal, whereas in Leishmania the body divides into longitudinal 2 pieces. Okay. Now moving forward to what is multiple fission. So multiple fission, you can see I've given the example of plasmodium. So under unfavorable conditions, whenever the conditions are not favorable, many daughter cells are stored inside a cyst. Okay, you can see I have given the diagram here. So you can see that a protective layer is formed around the cell to protect these daughter cells. Okay, the nucleus divides into daughter cells and it is stored. When the conditions are favorable, you can see that the cyst breaks. Okay, the cyst breaks down, making the daughter cells to come out. Okay, so you can see that in this diagram, you can see that the daughter cells are released in the favorable conditions. So under unfavorable conditions, a cyst is formed and then the cyst will protect. Okay, this cyst will protect the daughter cells, and then these daughter cells are released when the conditions are favorable. Example, plasmodium. Okay, so plasmodium, one more important point is to remember that it is a malarial parasite. Okay, very important point to note that this causes malaria. There are many species of plasmodium, but let us not go there. So this is a malarial parasite. So this question can be asked for 2 marks each. Okay, so whenever a diagram based question is asked, you are expected to write the statement about what is binary fission, define it, and then draw the diagram along with the labeling. Okay, now moving forward to what is fragmentation. The word fragmentation means splitting. Okay, splitting into 2 or many. Okay, so here you have to understand that fragmentation is the breaking up of body of simple multicellular organism into 2 or more pieces on maturing, each of which subsequently grows to form a complete new organism. Okay, the example is Spirogyra. So example of fragmentation is Spirogyra. So in Spirogyra, you can see that this is the parent Spirogyra, and after maturation, the filament breaks into 2 daughter Spirogyras. This further leads to the formation of a new Spirogyra organism. Okay, so this is how fragmentation takes place. What is the difference between fission and fragmentation? The difference is that fission takes place in unicellular organism, whereas fragmentation takes place in multicellular organisms. Unicellular organisms example is amoeba. Multicellular organism, you can give the example of Spirogyra. Okay. Now there is one more question that is asked. What happens when a mature Spirogyra attains a considerable length? So what happens? Spirogyra reproduces asexually by fragmentation. When a mature Spirogyra filament attains considerable length, it breaks into 2 or more fragments, and then these fragments then grow into complete organisms. Okay. Next, regeneration. Regeneration simply means formation of something new, right? So formation of new from the old. Okay. So in regeneration, basically, it is the process of getting back full organism from its body part is called regeneration. In case of Hydra and Planaria, you can see in the case of Planaria that the body of the Planaria gets cut, okay, at certain places, and each part that is cut grows into an individual Planaria. Again, this part will be cut and then it grows into a separate new organism. Okay, This is possible because of certain specialized cells called regenerative cells. Okay, this is possible because of certain cells called regenerative cells. In case of certain organisms like Hydra and Planaria, regeneration is a process of reproduction where a whole new organism is produced from a single body, okay, or single part of a whole organism. But in case of certain organisms like lizard, you might have observed that whenever a cut part, okay, whenever a cut part of the lizard, whenever a body of the lizard gets cut, it grows back. For example, the tail. So this is also a process of regeneration where only the body part, the lost body part grows. Okay, lizard, it could be starfish, okay, starfish, and then salamander. So all these are examples of certain organisms who have the capacity to regenerate the lost body part, and not the whole organism. Okay, so here in salamander, starfish and lizard, you have to understand that reproduction doesn't take place by regeneration. Here only the lost body part is getting regenerated. Okay, whereas Hydra and Planaria, only in those organisms regeneration is a process where reproduction takes place by the help of regenerative cells and a whole new organism is produced. I hope you have understood this. Okay. Next is budding. So in budding, small part of the body of the parent organism grows out as a bud, which then detaches and becomes a new organism. Example, Hydra and yeast. So you can see that from the parent, okay, so from the parent organism, a small outgrowth is growing, which is called as the bud. This bud grows and then it grows until it gets complete nutrition and then falls off as a new organism. It gets detaches from the body of the parent and then forms a new organism. For example, Hydra, and this is a part of your NCERT textbook, but yeast they have not given in your textbook. So I am just showing you the diagram here. This can also be asked for 2 marks. Okay, so please make a note of this diagram. So yeast, you can see that from the parent an outgrowth is growing, and this is called as a developing bud. Now this developing bud forms a new bud and then a chain of buds is formed, and when the conditions are favorable, this bud will get detached and grow into a separate new organism. Okay. Okay, so I hope you have understood what is budding. Next is spore formation. So it is very simple and understandable that spores are formed. So what are spores? Basically, spores are very light. They are the reproductive part of the certain fungi. Basically, you might have seen that whenever you place a piece of bread, okay, whenever you place a piece of bread outside by mistake, you see some black spots on the bread. Okay, this is nothing but the fungus bread mold. Okay, this is nothing but the fungus bread mold, also called as the rhizopus. The name of this bread mold is rhizopus. So what happens is, this rhizopus, when it attaches or when it gets attached on the bread which is moist, they release the spores which are present in these blob-like structures or balloon-like structures called as the sporangia. Okay, these sporangia are held with the help of filamentous thread-like structures called as the hyphae. Okay, so hyphae are the filamentous structures which are the non-reproductive part. Okay, they are the non-reproductive part. They don't help in reproduction, whereas they help in the holding of the blob-like structure called as the sporangia, which are the reproductive part of the fungus or this rhizopus. So these spores are very light and they float in the air and they are carried over large distances by air. Each spore is covered by a hard protective coat to withstand unfavorable conditions such as high temperature and low humidity. Due to this, spores can survive for a longer time. Okay, so these spores these spores are only let out by the sporangia when the conditions are favorable. When the bread is completely moist, you can see that spores are formed. They are released in the air. Okay, and these fall down on the bread. These fall down on the bread, forming the new set of sporangia and new set of organism. Is that clear? Alright. Moving forward to the next set of asexual reproduction, that is the vegetative propagation. Vegetative propagation is one of the asexual method of reproduction in plants, where we can see that there are many plants in which parts like the roots, stem and leaves develop into new plants under appropriate conditions without the help of reproductive parts of the plant. Now, what are the reproductive parts of the plant? They are the flowers. So flowers, which means fruits, seeds, okay, all these come under the reproductive parts. Basically, flowers come under the reproductive parts. So vegetative propagation is basically done in those plants which cannot produce seeds on their own, which do not have the capacity to produce seeds, okay, or even if the seeds are produced, they are not viable, which means they cannot fertilize further, okay. I hope it is clear. Now, under vegetative propagation, we have two types. One is natural vegetative propagation and artificial vegetative propagation. The example of natural vegetative propagation is bryophyllum. So, bryophyllum leaves, you can see that the leaf margin, this is called as the leaf margin. Okay. So, in the leaf margin, there are buds. Okay. There are buds formed, and these are called as the adventitious roots. Okay, the buds are formed and these buds will fall off when the conditions are favorable and form a new plant naturally. So this is a natural vegetative propagation. Under artificial vegetative propagation, we have examples such as sugarcane, rose and jasmine. So, sugarcane, rose and jasmine comes under artificial mode of vegetative propagation, and these are propagated using cutting, layering and grafting. So, like I told earlier, artificial vegetative propagation is done for those plants which produce seeds that are not viable, that are not useful to the plant, or they are not fertile. Okay, or those plants which do not develop seeds at all. Okay. Now, moving forward to artificial vegetative propagation. Artificial vegetative propagation, the first one is cutting. So, what they do, it involves cutting a piece of plant and rooting it to produce a new plant. Simply, a plant is cut. Okay, you can see it is cut, and this part of the plant is placed in the soil, and then it leads to the formation of roots, and then it grows into a new plant. Okay, this is cutting. Next is layering. Now, what does layering mean? What they do in layering is they take a branch, okay, for example, they take this branch of the plant and it is covered in the soil. So it is simply put in the soil, and once the roots start developing, this part of the plant is cut here and this grows as a new individual plant. Okay, this is the example. This is the process of layering. Next, grafting. Grafting is done for sugarcane. It occurs when two different plant stems are joined and they grow as a single plant. Now you can see that one part of the stem is from a different plant and the second part of the stem is from a different plant. Okay, these two are tied together and then this is called as grafting, and this grows into a new plant. Suppose let us take one part of the plant. Okay, this stem is of a apple tree or apple plant which has good apple fruits but is always prone to diseases. Okay, it is not disease resistant, let us say like that. And this part of the plant, this is another apple plant which is resistant to diseases. Okay, resistant to diseases. So when you keep these two together, you get a healthy plant which produces good apple quality and also it is disease resistant. So one part of the plant, so the plant part which is inside the soil is called as a stalk, and the one which is brought from another plant is called as a scion. Okay, this part is called as the one which is present in the soil is called as the stalk, and the one which is brought to get it attached or to get fixed into this is called as a scion. Okay, so this produces good variety of plants, this method of grafting. All right. Next is tissue culture. So what exactly happens in tissue culture? Tissue culture is basically where a plant is taken and the tissue is isolated from that plant and placed in a medium containing nutrients. So this is a medium containing nutrients such as agar. Okay. Now, after keeping the tissue isolated from the plant in the medium, you can see the formation of callus. Callus is nothing but new cells that are formed. Okay. Now this callus again is transported into another medium containing hormones such as auxins. Okay. So when you place the new cells in auxins, they help in the promotion of growth of the shoot and the root, and saplings are obtained. Now these saplings which are obtained are placed in the nursery bed or the field. Okay, and then it is grown and then yield is obtained. So I hope you have understood the whole concept of asexual reproduction in plants and animals. In the next class, I'll be teaching you sexual mode of reproduction. So if you have any doubts regarding this topic, please do post it in the comment box and like the video if you have understood the concept thoroughly. Subscribe to my channel if you have not subscribed yet. Okay? And as I always say, stay positive. Stay motivated. Thank you.